In this video, we're going to look at what makes up blood and the importance of some of these components in haemostasis, which is the normal response of the blood vessel to injury by forming a clot that serves to restrict the amount of blood lost through the wound. What we are looking at here is a stained blood smear, where a drop of blood has been placed on a clean microscope slide, and then a second slide used to draw the blood across that slide to produce a sample that is a single cell layer thick. The cells in the blood can then be stained to allow those cells to be viewed under a light microscope. And what we can see in this field of view are some leukocytes or white blood cells. So we have a couple of neutrophils and a mononuclear cell. Then we have a lot of red blood cells or erythrocytes, which are the most abundant cell in the blood. And then these small dots, which are our platelets, and despite being tiny, play a fundamental role in our survival. Now, our blood is made up of a wide range of different components, and this diagram shows what a blood sample looks like if it is centrifuged to separate some of those components. The top layer, this straw-coloured liquid, is the plasma, and plasma makes up about 55% of the blood volume, and it's rich in proteins that can be broadly divided into three main classes: albumins, globulins, and clotting factors. It also contains a range of inorganic ions, such as sodium and chloride. Just below this, we have a cloudy white layer, which we call the buffy coat layer, and this contains our leukocytes or white blood cells and platelets. Finally, the red blood cells or erythrocytes are the densest component of blood and form this bottom layer. The buffy coat layer and red blood cells together are called the formed components of blood and make up about 45% of the blood volume. This is what a real sample of blood that has been centrifuged looks like, and we can make out those three distinct layers. For the average adult human, blood makes up approximately 7 to 8% of the body mass. So for a 70 kilo adult, they will have a total blood volume of approximately 5 litres. What about the functions of blood? Well, the obvious one that most people would think of first is as a transport mechanism for oxygen. But it also transports waste products of cellular metabolism, for example carbon dioxide or urea. It allows for the movement of lipids, as well as hormones and other cellular communication proteins, whilst distributing soluble nutrients like vitamins and glucose around the body. The blood also plays a central role in host defense. It allows leukocytes to move around the body. It carries antibodies in the plasma, as well as proteins of the complement system. And although not strictly a defense mechanism, it helps regulate our body temperature by carrying heat to the surface of the skin while maintaining a constant central body temperature, allowing all of these processes to function more effectively. If you've watched a video on the immune system, then you may have seen this diagram before, which is the different cells that can be produced from hematopoietic stem cells, and we focused previously on the leukocytes or the white blood cells. In this video, we're going to look briefly at the red blood cells and then the role of platelets in haemostasis in much more detail. The erythrocytes occupy the largest volume in the blood, with approximately 5 trillion red blood cells per litre of blood. The hematocrit is a measure of how many red blood cells there are per litre, and involves calculating the ratio of red blood cells to total blood volume per litre. Normal values for adult males are about 47%, and slightly lower in adult females at about 42%. The higher the hematocrit, the more viscous the blood will be. The primary role of erythrocytes is the transport of oxygen from the lungs to tissue and the removal of carbon dioxide. And red blood cells have a lifespan of about 120 days, after which they are removed from the circulation in the spleen, with the overall levels of erythrocytes being under the control of a peptide hormone called erythropoietin, or EPO. And you may have heard of this in the context of doping in sport, as it allows the body to produce more red blood cells, thereby giving athletes a competitive advantage as they can transport more oxygen around their bodies. Moving on to platelets, which are also known as thrombocytes. Platelets are absolutely essential to life as they help control bleeding and maintain the integrity of the vessel walls or the vascular endothelium. They are not strictly cells at all, as they are fragments of a much larger cell called a megakaryocyte. The megakaryocyte forms these protrusions from which platelets bud off and form tiny fragments that lack a nucleus but do contain granules, which are really important for their role. They may also contain mitochondria and small amounts of endoplasmic reticulum, and each megakaryocyte can give rise to around 1,000 platelets. Platelets only have a lifespan of about 10 days, and as mentioned, they are rich in granules. They have 3 different classes of granules with content that plays a key role in haemostasis, as we will see when we describe the clotting process in more detail. Alpha granules are rich in platelet-derived growth factor, or PDGF, von Willebrand factor, or VWF, and various clotting factors including fibrinogen. Delta granules contain calcium, ATP, ADP, and 5-hydroxytryptamine, which is another name for serotonin. Finally, the lambda granules are rich in hydrolytic enzymes. What we can see in the electron microscope image on the right is an activated platelet, which is forming these protrusions called filopodia, and these are really important for their function in being able to cross-link with one another. We've mentioned the process of platelets and haemostasis, but what exactly does that mean? Well, hemo refers to blood, and stasis to slowing down, so it is literally the localized slowing down of blood flow, which occurs in response to mechanical injury of some kind to the blood vessel walls. Haemostasis is a coordinated sequence of 5 steps: vasoconstriction, platelet aggregation, clot formation or coagulation, vessel repair and clot retraction, and fibrinolysis. Before we dive into the details of each step, it's important to appreciate why blood does not clot in the absence of damage under normal physiological conditions. And to understand this, we need to briefly look at the structure of a blood vessel and its underlying layers. A healthy blood vessel has a lumen, which is made up of the endothelial cells, surrounded by layers of connective tissue, formed from collagen fibers and elastin, with a layer of smooth muscle around the outside. If we zoom in on the structure of the inner layer a little bit, so now we are focusing on a healthy endothelial cell layer, which makes up the vessel wall. And these cells and the components they secrete or express on their surface are central to regulating normal blood flow. Blood in our circulation is maintained in a fluid state, and inappropriate clotting is controlled by 3 primary mechanisms. The endothelial cells themselves are secreting nitric oxide, or NO, and prostacyclin, PGI2, which keeps platelets in an inactive state and prevents them from binding to the endothelium. The endothelial cells also express heparin sulfate on their surface, which is a natural anticoagulant, but this can enhance its anticoagulant effect by binding a molecule called antithrombin, which you may also see called antithrombin 3, but which degrades or breaks apart clotting factors 2, 9, and 10, which we will discuss in a moment. Lastly, thrombomodulin binds thrombin or clotting factor 2, which in turn activates protein C, resulting in the activation of clotting factors 5 and 8. So now that we understand what happens normally, let's look at what happens during the different stages of haemostasis. Vasoconstriction occurs any time there is mechanical damage to the vascular wall and underlying structures to prevent excessive amounts of blood from escaping the blood vessel. The damaged endothelial cells release endothelin, which binds to the underlying smooth muscle cells, causing them to contract, which in the short term reduces the amount of blood flowing to the area and therefore escaping the vessel through the site of damage. To further enhance this vasospasm, we have a mechanism called myogenic contraction, which is a spontaneous contraction of the smooth muscle cells due to an outside stimulus, such as the damage caused to the underlying tissue. Inflammatory mediators, including histamine, leukotrienes, and prostaglandins, are able to bind to nociceptors or pain receptors, again inducing smooth muscle contraction. So we have 3 mechanisms to induce vasospasm or vasoconstriction, but this is just a short-term fix, and the next stage of haemostasis is the formation of the platelet plug. Let's zoom in a little bit on the damaged area so we can see what is happening in a bit more detail. As the damaged endothelium is not releasing nitric oxide or prostacyclin, the platelets are able to become activated, forming these filopodia, and within seconds of the injury occurring, these activated platelets start to accumulate around the damaged endothelial walls, adhering via integrin receptors facilitated by von Willebrand factor or VWF, which is released initially from the damaged endothelial cells. The activated platelets degranulate, releasing their contents, including ADP, thromboxane A2 or TXA2, and 5-hydroxytryptamine, which is also known as serotonin. This induces a positive feedback loop as the ADP and TXA2 bind to circulating platelets, activating them, and recruiting them to the site of damage, rapidly increasing the formation of the platelet plug, which blocks the hole in the vessel wall and stops more blood from leaving the circulation. The platelets are held together by a combination of von Willebrand factor and fibrinogen, which cross-links platelets' filopodia and helps provide some stability to the otherwise unstable structure. Thromboxane A2 and serotonin bind to the underlying smooth muscle cells, triggering further sustained contraction of the vessel walls to enhance the vasoconstrictive effect. The third step of haemostasis is coagulation. We have formed this platelet plug, but the structure is still quite unstable and could easily be dislodged. So the next stage of the process is to stabilize the plug and blood coagulation. Even when we do not have any vessel trauma, the liver is producing high quantities of different clotting factors. A few we have already mentioned when looking at the protective mechanisms that prevent inappropriate coagulation. But these factors are constantly circulating in the blood in an inactive form or as zymogens, ready to become activated within seconds. The clotting cascade requires the sequential activation of clotting factors from their inactive precursor form to their active form by the action of a previously activated clotting factor, as shown in this diagram. Many of the activated clotting factors are serine proteases, and we denote the activated form with the small letter A. So in this example, factor 10 is being converted from its inactive precursor to the active form, factor 10A. There are two pathways that become activated during coagulation, the intrinsic and extrinsic pathways. Looking at the intrinsic pathway, which is so named because all the components needed for its activation are present within the blood. The first clotting factor to be activated is factor 12, which interacts with the negatively charged surface of the platelets and converts itself to the activated form 12A. 12A in turn activates factor 11, converting it to 11A. 11A sequentially triggers the activation of factor 9, forming factor 9A. Factor 8 interacts with the activated factor 9 and forms a complex that also requires the binding of platelet factor 3, which comes from the cell membrane of platelets, and calcium, which is released from the delta granules of the activated platelets. This complex converts inactive factor 10 into its active form, which is the first stage of the common pathway. Factor 5 associates with factor 10A, and this 10A5A complex binds a protein called prothrombin activator, which converts factor 2 into the active form. Factor 2 is also called prothrombin, and the active form, 2A, is thrombin. Thrombin is one of the central proteins in the coagulation cascade and interacts with circulating soluble fibrinogen, or factor 1. When thrombin interacts with the fibrinogen molecules, it polymerizes them, forming an insoluble molecule called fibrin, which is factor 1A, and this causes the blood to become more viscous or gel-like. Factor 13, or fibrin stabilizing factor, becomes activated by thrombin in the presence of calcium, and this factor starts to cross-link the insoluble fibrin strands, start forming a fibrin mesh or net-like structure. This fibrin mesh covers the platelet plug, stabilizing it and preventing the plug from dislodging. And just to note, all of these reactions are actually occurring on the surface of the platelet plug. Now, this pathway we've just described is the intrinsic pathway, which occurs inside the blood. There is a second pathway called the extrinsic pathway, which is triggered by tissue factor, or factor 3, which is released from the damaged endothelial cells. Factor 3 activates factor 7 to 7A, which has two effects. It can enhance the conversion of factor 9 in the intrinsic pathway, but it can also converge with the common pathway, which is anything from factor 10 onwards, as we have already described for the intrinsic pathway. The advantage of the extrinsic pathway is that it is incredibly quick to become activated, often within about 30 seconds, whereas the intrinsic pathway, because there are many more proteins involved, can take around about 5 minutes to become fully activated. What's really neat about this cascade, though, is that there are positive feedback loops built in. The downstream activation products can act back on earlier stages of the cascade to amplify the cascade effect and drive a really effective clotting process. Okay, so once the platelet plug has been stabilised and we are no longer losing blood, we need to repair the damage to the vessel wall and then retract the clot. So the platelets in the platelet plug have contractile ability due to the presence of actin and myosin fibres. And the platelets contracting brings the damaged edges of the endothelial cells closer together, forcing the serum that is trapped in the platelet plug out into the circulation. Serum is basically plasma that does not contain any clotting factors. At the same time, the platelets release platelet-derived growth factor, which acts on the underlying smooth muscle and collagen to produce tissue patches, which drives the repair process for these underlying structures. Lastly, the platelets secrete vascular endothelial growth factor, or VEGF, which acts directly on the vascular endothelial cells to repair any damage and regenerate the vascular lining. The fifth and final step of haemostasis is a process called fibrinolysis. Once the vessels and underlying structures have been repaired, we need to get rid of the clot so that it does not impede blood flow. Tissue plasminogen activator is present on the surface of all endothelial cells, and it cleaves a protein called plasminogen, forming plasmin. The plasmin molecules start degrading the fibrin mesh, which breaks apart the clot and prevents any occlusion of the blood vessels. This tissue plasminogen activator can be used therapeutically in stroke patients to help break apart clots that might be blocking blood vessels in the brain. And the activation of plasmin can also be driven by kallikrein, which is a serine protease enzyme. So that covers the 5 stages of haemostasis. And just to recap, the first step is vasoconstriction or vasospasm, which helps prevent blood loss. Secondly, platelet plug formation to start sealing the damage to the vessel wall. Thirdly, coagulation, which results in the formation of the fibrin mesh, which stabilizes the platelet plug. Fourthly, clot retraction and repair, which draws the damaged endothelial cells together and drives the repair of the vessel walls and underlying structures. Fifthly, and finally, fibrinolysis, with the production of plasmin, which degrades the fibrin mesh, breaking apart the clot and allowing it to be flushed away to restore normal blood flow. Haemostasis is an essential process in the body, ensuring that we do not lose too much blood due to trauma and effectively repairing the vessel walls to restore homeostasis. The regulation of haemostasis is equally important, helping to safeguard against inappropriate clot formation, which could cause medical complications.